invasive plants, mammals, reptiles, and amphibians. Compared to insects and birds, the effect of invasive plants on habitat quality for mammals, reptiles, and amphibians has received comparatively less attention. This presentation summarizes the results of what research is available. Research on the effects of invasive plants on small mammals has found that in areas where honeysuckle dominates the forest understory, food and cover opportunities increase for small mammals, leading to greater small mammal abundance in these invaded habitats. In a bit of a feedback loop, the greater abundance of small mammals results in greater predation on native tree seedlings, which provides the honeysuckles with a competitive advantage and further enhances the invasibility of honeysuckle. In western U.S., invasive cheatgrass grows denser than native grasses and significantly increases the density of the herb layer in sagebrush ecosystems. This has the effect of reducing small mammal mobility and foraging efficiency, which leads to a reduction in the number of rodent species that use invaded areas as habitat. In New England, most remnant populations of New England cottontails occur in very small habitat fragments that are dominated by multiflora rose and autumn olive. Compared to most native shrub species, multiflora rose and autumn olive provide cottontails with superior cover from predators, and multiflora rose in particular provides excellent winter cover because the large arching structure of these plants trap the snow above the ground and form air pockets under the shrubs that rabbits use as winter dens. New England cottontails face many challenges. The remaining animals occur basically in islands of dense shrubby habitat surrounded by a very inhospitable landscape that lacks shrubby cover and is full of predators that love to eat cottontails. We find that most New England cottontails remain within their habitat patches because when they try to leave that patch of cover, most rabbits get gobbled up by predators. Although most invasive plants seem to be poor quality browse for cottontails, and cottontails residing in dense patches of barberry seem to be exposed to very high tick loads, biologists seem to agree that in most habitats, if we remove the invasive plants, we would effectively result in losing the cottontails from those habitats. Similar to cottontails, deer typically avoid browsing most invasive plants unless other foods are very scarce. Our landscape is like a salad bar for deer, and they move across the landscape, browsing the native plants they prefer and avoiding the non-native plants that taste bad to them. This selective browsing of native plants by deer, especially when large numbers of deer occur within a landscape, gives non-native plants a competitive advantage and is a significant factor that contributes to some plants becoming especially invasive. For example, deer are believed to facilitate the invasion of non-native garlic mustard and stiltgrass by reducing the cover, height, and reproduction of native plants. This has a cascading effect to other wildlife because where browsing by deer encourages stiltgrass invasion, forest songbird diversity has been shown to decline. Deer not only help invasive plants by browsing competitive native plants, deer disperse invasive plant seeds in their fecal pellets. A single pellet group, one defecation, may contain over 300 honeysuckle seeds that have a germination rate as high as 80%. In landscapes where deer density is high, reducing deer density via hunting may be the most effective way to encourage native plants. For amphibians, Japanese knotweed has been found to reduce the diversity and abundance of invertebrates, primarily because it grows tall and dense and reduces overall plant diversity. This leads to a reduction in the quality of foraging habitat that green frogs use in the fall prior to hibernation. Honeysuckles can exude chemicals into temporary wetlands, and these chemicals have been shown to reduce the respiration and survival of American toad tadpoles. However, spotted salamanders and 2 other frog species breeding in these same wetlands suffered no negative effects. Invasion of dense stands of common reed, Phragmites, can reduce the amount of open water in marshes, and this has been shown to lead to a decline in Fowler's toad populations in Ontario. And by growing especially dense, common reed can significantly reduce the number of amphibians and reptiles that will use invaded areas as habitat. Blanding's turtles typically avoid dense patches of common reed, probably because they are difficult to move through. The dense cover and shade reduce foraging opportunities for turtles, and the shade creates poor thermal conditions for turtles. When turtles don't avoid areas of dense common reed, they can get stuck and desiccate. Shading by common reed has also been found to increase the length of time turtle eggs require for incubation, and in some cases, the stems of common reed have been documented growing through the eggs in turtle nests, further reducing hatching success. If you have any questions or comments about this presentation that you would like to share, please email those to me at matt.tar@unh.edu.